Hi everyone, I'm Naomi. I worked with Globus Labs over the summer, and my project was on adaptive tool selection in a scalable genomics pipeline. So for some background on my project, it was based off of an FMT viral detection pipeline that I created as part of my undergraduate research at U of A. Throughout the summer, I used Parsl to make my original pipeline more efficient and also scalable. And then from there, I applied adaptive tool selection with Academy. So some background on FMT viral detection. FMT stands for fecal microbial transplant, and this is a procedure that's used to treat C. diff. C. diff is a bacteria that grows in somebody's gut and basically overgrows and disrupts the balance of all good bacteria in that person's gut. So one way to heal this type of infection is through a stool transfer. So they transfer stool from a healthy donor to a patient's digestive tract. And then within that stool, there's all of the bacteria that is needed to regrow the patient's gut microbiome. And through this project, I'm not looking at bacteria as they transfer from donor to patient, but I'm looking at viruses and what role they might play. And a lot of the viruses I'm looking for are temperate viruses, meaning that the genetic material is inserted into usually a bacterial host cell's genome. So I'm using metagenomic data. And that's why I can use the same data from this diagram here, which came from the paper that I got my data from. And this diagram is looking at bacteria as they travel from donor to patient, and then also over time. So what bacteria stick around in the patients as their gut heals. So my project is very similar, except I'm looking from viruses and what viruses transfer from donor to patient, and then also how they stay over time. And my workflow takes in assembled contigs. So there's some pre-processing steps that needs to be done, and creates a list of all viruses found within all of the samples, along with their annotation. And from there, they have sample IDs that can be traced back to, did it come from a donor or a patient, and at what point in time. So this is what my original pipeline looked like. It was formatted as individual shell scripts for every single step. And then there was one launch script that launched every shell script off with sbatch. And as you can see, each step can run each sample in parallel. However, every single sample has to be done with one step before the next step can start. So with Genomad here, this is the actual viral detection step. Every single sample has to be done running with Genomad before check-v, which is the quality step, can be run. And so from here, I ported this pipeline into Parsl. And Parsl allowed me to make my workflow a lot more efficient. So instead of having to wait for every single sample to be done with Genomad, each sample can go through these first 3 steps on its own without having to wait for every single sample to be done with one step. And as you can see, before the clustering step, we still have to wait for all the samples to be done. This is just due to the nature of how the clustering works. The clustering takes all viruses that were identified in the samples and combines them into one file so that it can find representative sequences, just because there will be so many sequences identified. But by still being able to parallelize and make these first 3 steps more efficient, it actually cut off a lot of time from our original workflow because these first 3 steps were the most time-consuming steps of the workflow. And so this is what it looked like when we did some scaling experiments on the workflow after it was ported into Parsl. As you can see here, the original sequential workflow was almost at 4 hours. And after scaling this up, after it had been ported into Parsl, at about 4 nodes for 3 samples, it hits its maximum efficiency at about 2 hours. That's almost a 50% decrease in time needed. And it kind of gets less clear past 4 nodes. And this is most likely just due to there not being enough computing that actually can fill more than 4 nodes, which is why we tried it out with 12 samples. And with 12 samples, we saw that it scaled even farther, a little bit farther to like 6 nodes, and the time again to about 2 hours from originally almost 9 hours. So as you can see, with more samples, it will scale a little bit farther, and the original data set had about 110 samples that we could scale. And after scaling and porting the workflow into Parsl, I agentified the workflow. So originally it was a linear workflow with different steps in order. And this is more of a looped workflow. So every single step that was there originally is now an agent, and each agent is individually ported into Parsl here. And by doing this, I'm allowed to have I am able to have different configurations for every single agent, which was really helpful. So for example, the viral detection agent requires a lot of computing resources, whereas the CheckV agent or the clustering agent just needs a lot of memory. So I was able to customize those things and make my resource use a lot more efficient. Another difference here is that instead of just using Genomad to detect viruses, it's now a viral detection agent. So there's three different tools. There's VirSorter 2, Deep VirFinder, and Genomad. And these tools all differ in whether they are homology based, so comparing the findings to a database, or machine learning based, so looking at patterns. And how this agentic version of the workflow works is that the CheckV agent, which checks for the quality of the viral findings, sends information back to the viral detection agent about how high quality its findings were. And the BLAST agent, which matches the viral findings to a database and collects annotation, sends a score back to the viral detection agent about how many viruses actually match to the database. And from these two scores, the viral detection agent can dynamically choose which tool might be the best tool for this data set. So for example, if a data set had a lot of commonly found viruses, a homology-based tool like VirSorter 2 might work best. If there was a data set with a lot of rare viruses, a machine learning tool like Deep VirFinder might work best. That's my project though. If you want to look at my code, there's a link and also a QR code. But thank you, everybody.